Welcome to this video protocol where we will take a step into the exciting field of spatial transcriptomics. Here we will describe how we prepare formalin-fixed, paraffin-embedded prostate cancer sections for spatial transcriptomics. The polyclonal evolution of prostate cancer poses a challenge for personalizing treatment. Here we illustrate predominant intermediate-grade disease. With progressive clonal evolution, subclones are formed, some with the capability to metastasize to distant sites such as lymph nodes and bone, underscoring the complexity of disease progression. Leveraging samples from prostate biopsy, prostatectomy specimen, and lymph node metastases, we aim to map and characterize genetic features of clonal lethality. At the Oxford Prostate Cancer Biology Group, we have developed an expertise in interrogating prostate cancer heterogeneity through spatial transcriptomics. Our seminal publication in Nature utilized fresh frozen prostate tissue. Taking the next step to leverage the breadth of archived tissue, we've developed and refined an approach to use FFPE specimen for spatial transcriptomic evaluation. Starting with the preparation of the prostatectomy specimen, its journey begins following a robot-assisted radical prostatectomy. The resected specimen is first fixed in neutral buffered formalin overnight to preserve its histological architecture. Following inking to preserve orientation of the specimen, the seminal vesicles are removed and the sectioning of the specimen commences. Initially, the apex and base of the prostate are sectioned axially and put aside. The body of the prostate is then axially sectioned. Here, we can appreciate the tumor macroscopically in the anterior and left lateral zones of the prostate. The apex of the prostate is then sectioned sagittally and placed in a separate embedding cassette. The apex specimen is sectioned orthogonally in a sagittal plane compared to the axial plane used for the body of the prostate. This is repeated for the base of the prostate, and the sections are subsequently transferred to separate embedding cassettes. The prostate specimen has been systematically sectioned and is now ready for embedding in paraffin. There are three key stages in preparing a FFPE histological specimen for spatial transcriptomics. First, the specimen is stained with hematoxylin and eosin. The stained section has to be digitalized and annotated by a pathologist. Next is stage 2, which is digital planning. Here, a schema as shown on the right is overlaid digitally to plan sectioning of the tissue. In this example of a prostatic apex specimen, to achieve maximum sequencing coverage of region of interest, we have planned two regions for spatial transcriptomic sequencing outlined in the red square, which will be on separate slides. Stage 3 involves sectioning of the FFPE specimen. With our digital planning complete, we take the next step where we directly superimpose the digital schema onto the FFPE block. The dotted line here represents the incision line for dividing the section to be mounted and processed for sequencing, which will be on separate slides. Here, we demonstrate scoring the incision line on the FFPE block using a scalpel. These scores will allow for standardized and efficient separation of sections for mounting on separate histology slides. Using a microtome, 5 micrometer sections are cut from the FFPE block at room temperature. The sections are carefully transferred to a water bath which is kept at 45 degrees Celsius. Here, we illustrate how the score incision on the FFPE block allows for precise separation of the section according to the digitally planned schema. The sections are carefully mounted on a superfrost plus gold slide. It is important to ensure the section is carefully oriented in the middle of the slide, with the tissue oriented according to the planned schema. Shavings from respective regions of interest are also taken and placed in an Eppendorf tube for DNA fluorometric analysis. This is part of our quality assurance steps. By following this video protocol, you should achieve slides as shown here, reflective of the digital schema, optimized for spatial transcriptomic sequencing. Next, we will move on to the preparation of prostate biopsy and lymph node specimen. The linear structure of prostate biopsy cores require more extensive planning and technical expertise when mounting to ensure optimal sequencing coverage. This illustrates a set of biopsy cores which have been mounted, ready for subsequent processing. Here we illustrate the digital schema for various H&E stained iliac lymph nodes containing prostate mets. These sections will be prepared for sequencing as previously described. After staining and decross-linking to overcome protein crosslinks from formaldehyde fixation, the slides are loaded onto the Visium Cytassist platform to facilitate RNA capture on spatially barcoded Visium slides. Now, your sections are ready for spatial transcriptomic sequencing. In the next part of this video, we'll explore a range of challenges we faced when preparing FFPE section for spatial transcriptomics. As a bonus feature, we've also included solutions to overcome them. For larger sections, such as the prostate axial disc, with multiple regions of interest to be mounted on separate slides, the sections can be carefully separated along the incision scalls using a forceps when in the water bath. Keeping the FFPE block on ice during preparation and in between sectioning can help reduce the incidence of creases. Ensuring these sections are not kept on the hot water bath for far too long can help reduce the formation of cracks. To prevent lifting of sections on subsequent processing, the use of charged slides and ensuring no air bubbles are present when floating sections on the water bath can help. Bringing it all together, from the prostatectomy and lymph node specimen, along with our comprehensive bioinformatic pipeline to process spatial transcriptomic data, we can reliably interrogate the prostate cancer clonal landscape. This opens up a wealth of interesting translational questions that can improve personalization of prostate cancer management. We at the Oxford Prostate Cancer Biology Group are excited to share this video protocol with the wider scientific community. This work is part of the SPACE study, where we are uniquely positioned with our expertise on spatial transcriptomics to identify the lethal clone within primary prostate cancer.